Hello guys, today we are going to discuss about a lab test. It is called bacterial wound culture test. What is the use of the test? To detect a bacterial wound infection, to determine which specific bacteria are present, and to isolate and grow the bacteria for a subsequent susceptibility test. A bacterial wound culture is a test that is used to detect and identify pathogenic bacteria in a potentially infected wound. Wounds may be superficial breaks in the skin, such as scrapes, cuts, scratches, or may involve deeper tissues such as incisions, bites, punctures, or burns. Any wound may become infected with a variety of bacteria. A culture helps to determine which type of bacteria are causing an infection and which antibiotic would best treat the infection and help heal the wound. A culture is performed by collecting a sample of fluid or cells tissue from the wound and placing it on or an appropriate nutrient media. The media encourages the growth of a bacteria that may be present, allowing for further testing and identification. Typically, if a person has a wound infection, there will either be a pure culture of the microorganisms, only one kind will be found, or one type will predominate. In some cases, for instance with a human or animal bite, there may be several pathogens present. Since wounds may be superficial or involve deep tissues, they may harbor different types of bacteria that have different requirements for growth. Some bacteria infecting a wound may require air for growth, aerobic, while some require a non-oxygen or no oxygen, reduced oxygen environment called anaerobic. Care must be taken when handling the samples so that their growth is encouraged and the probability of their detection and identification are optimized. The next step in the process is to identify the different types of microorganisms present. Identification is a step-by-step process that may involve many tests and evaluations performed on the bacteria found growing in the culture. One such test, the gram stain, involves smearing individual colony types onto glass slide and treating them with a special stain. Under a microscope, the bacteria can be classified into gram-positive and gram-negative organisms, and by shape into cocci or rods. With this information and additional biochemical tests, the types of bacteria present can be identified. For many of the pathogens identified in the wound culture, antimicrobial susceptibility test is required to guide treatment and to determine whether the strain of a bacteria present is likely to respond to a specific antibiotic. In order to do this, a pure culture of the identified bacteria must be available, which may require additional time in the laboratory to separate and to identify each bacterial species. The wound culture, gram stain test, and susceptibility testing all constitute to inform the doctor which pathogens are present and what antibiotic therapy is likely to inhibit their growth. Sample: usually a sterile swab used to collect cells or pus from the site of suspected infection. Occasionally, aspirations of a fluid from a deeper wound into a syringe or a tissue biopsy may be required. Normally, the result should be sterile. That's all for today's test.